We're going to now move to the last section of today's event, and that is open it up for questions from the audience here and any questions that people may have who are viewing this online. And again, it's the online email is ask@gbsi.org. So I guess we could start in the room here, and you would have to use a microphone so that people can hear. Who would like to ask the first question? Thank you. And then I will assign it to unless you have a burning desire to answer it.
Hi, Mike Herta from NIH, and I just want to bring to your attention something that we're working on that's very highly related called the Big Data to Knowledge Initiative, and part of that is very focused on standards with regard to data mostly, so probably worth keeping your eye on the website.
Thank you.
Ray Cypus, Chairman, President of ATCC. So I think when I envisioned the idea of the Global Biological Standards Institute, I moved from vision to implementation pretty quickly. And I think if there was one policy that I would encourage is that there would be a citation to a reference to standards in grant proposals, publications, and even at the level of talking to your mentor about the research you're trying to do. We should build that behavior immediately into the process. I think the second thing is if you look at the case history of standards, the successes, and I think you'd look at the Intellectual Property Organization, WIPO, and what they did, and if you look at the type culture system, which is a type of standard, in each case you had a governance structure. So I think what is very, very important in trying to implement success is a distributed governance structure where the three parts of the triangle that we mentioned, okay, would all be nodes or gatekeepers in the standardization process. And I think if they required reference to standards, okay, at each point, then you would get a system, an integrated, harmonized system among the various elements. And we have shown that that works. It is successful. So we know this is not fiction. This could be a real system that works. Thank you.
Any comments from the panel? Okay, thank you. We have an online question.
Hi, we have an online question that I'll read out. It is actually for Mary Lou. Somebody wrote in and said that you mentioned that it costs $10,000 to $12,000 to do a paper standard. The question is, what is the cost for the laboratory or scientist to comply with the standard? It must require changes in infrastructure, staff training, equipment, and they note that they agree that it is worth it, but wondering if there's an estimate of how much it costs.
That's an interesting question, and I don't have a good answer because it depends on where a laboratory is starting. If they already have a quality system in place, they don't go through any expense related to that. If they don't have a quality system in place, then it's going to be going through establishing their quality system, the time it takes to develop standard operating procedures that are going to be followed, et cetera. Most clinical, in fact, all clinical labs here in the U.S. are used to doing daily quality control. So they already have many of the elements related to standardization in place. Where they may not have it would be if they need to do things like establish normal ranges. And there's definitely going to be differences in the results you get depending on how you do a reference range study. That's one of the areas that CLSI has been very involved in. So if they aren't set up to do that, they're going to have to acquire that information and then put in place the documents that they need. So the bulk of it is going to come in terms of staff time to develop procedures. They're already paying in most cases for quality control materials, so there would not be added changes there. They may need to look at doing some internal auditing to make sure that staff on all shifts are doing everything according to whatever procedures exist. So it's going to vary a lot depending on where the institution is starting out.
And the person who wrote in, thanks you.
Steve Kaminsky, the International Rett Syndrome Foundation. Bill's statement reminded me of this. I think there's actually another challenge, and the challenge is education. I think that when funders, the NIH or International Rett Syndrome Foundation, fund researchers, and if they're funding, as Bill articulated, with a DoD mindset of 6.1 research, which is very basic research, the challenge, and as Len pointed out, with industry's actually thinking way down the road in technical readiness levels, and please review what technical readiness levels are. They're looking at all aspects of the technical readiness levels that the federal government actually put in place. But academics really only work in traditional technical readiness level 1. And asking an academic to go from technical readiness level 1 to technical readiness level 4 or 5 is actually a very, very large step. And I think there has to be a fundamental education in academia about how you move from a basic concept to all of the other aspects that the technical readiness levels articulate. Those are already out there. So I think it's a skill set, and it's an education of a skill set that needs to take place. And until we do that, we're going to constantly have this large gap, those two pieces of the bridge that don't meet. So I think at the laboratory level, that education has to take place. So if GBSI wants to lead, I think it's not only coming up with material standards, but educational standards in the laboratories at the level of undergraduates, graduate students, postdocs, and make sure that they get it, because the sooner we train them, the more we're going to see them producing the kind of results that our for-profit partners can actually move quickly with. As it is now, that partners can't move with the data that's coming out of the academic laboratories.
Steve, I could not, I'm sorry, go ahead, Bill.
No, go ahead. So
I was just going to say I could not agree with you more.
Yeah.
GBSI is discussing those kinds of education initiatives ongoing, and you couldn't have put it any better. Bill?
I just want to echo, I think you've hit a really perfect spot there. One of the things that a lot of universities do very poorly is translating ideas into marketplace products. Almost everyone is really pathetic in that view. The National Science Foundation started I-Corps. It's a program to essentially do that, to really actually get students exposed, faculty exposed to moving things forward. Another thing that's behooves industry to be involved in this whole web as well is to engage the academic community in ways that's they're not currently doing that. So a large part of what, at least my own experience at Maryland, we're trying to develop a nice ecosystem that involves fundamental research, companies, because we're next to the FDA and we have the CERSI and NIST, we have a very nice ecosystem that's building so that people that are involved in the 6.1 innovative idea research are also being exposed to what it really takes in that research, what results in that research companies need. Is it going to be patentable? Yes or no? Is it going to be standardizable? Yes or no? Can I take this and move it to the marketplace? Yes or no? And until you have that exposure to those problems, you're not going to get there. So I totally agree that a significant component is education, and that it's happening, but not to a significantly large extent.
Thank you. I think we have another online question.
Yes, we do. So the question comes from another online follower, and it is, I attended a talk not too long ago where the speaker argued that outsourcing of preclinical studies to contract research organizations, CROs, which he said is becoming increasingly prevalent, could help solve the reproducibility problem. Pay 2 CROs and you have reproducibility built in. Would you agree, or do you think people will just end up going to the CRO that gives them the answers that they want?
So I'll try and address that. To be clear, I spent more years than I like to remember, 25 years in academic research, and the last decade in industry. The truth is, I trust the results more coming out from a CRO than I do from an academic lab. The reason for that is typically when dealing with a CRO, you're able to set the boundaries of the study. And I've said already, blinding is the most important. And it's typically the case that a CRO will do the study in a blinded fashion. It's also typically the case that the CRO has less personal investment in the project than a postdoc or a student. The postdoc or the student has a lot of pressures in place to encourage them to get a positive result, which the CRO does not share. So I think that there's a lot to be gained by using CROs. Having said that, some of the research techniques and so on that are being used at the moment are so sophisticated and specialised, it's probably not reasonable to expect that the average CRO is able to handle those sorts of specialist techniques and doesn't have the appropriate expertise.
And I'll respond to that question as well. From my point of view, again, my background is industry, in the medical device industry. I would prefer to outsource to either a high-quality academic lab that I know will follow a protocol that is familiar with CLSI standards and will do things according to CLSI standards, or to a CRO, frequently than relying on in-house personnel. Because again, it's when you go outside, away from the people who develop the technology and are familiar with all the little things they do that they never capture in a procedure, that you're going to find errors or things that can't be reproduced. And I've seen this happen more than once. So I would always prefer to outsource. I don't necessarily go to CROs versus high-quality academic labs, but not all labs, whether they're CROs or academic labs, are equivalent.
I'd just also like to make a comment. I kind of alluded to this in my presentation. As pharma in particular they have for now several years, downsizing their own basic research discovery apparatus. They're using more CROs, but they're also looking to academia to carry out their own discovery basic research. And I think there's a cautionary flag being flown here that academic research has to be reproducible to the point where a large pharmaceutical company can trust that work if they're going to invest $500,000, $1 million, or more dollars in the University of X to do the kind of exploratory research that their own in-house teams no longer can do because they're not there. And I think that there's a danger there. And so I think that it behooves academia. And I've actually, as we were just starting GBSI, I've spoken to some very senior people at some very large pharmaceutical companies who have already invested several $1 million in academic collaborations, and they're very concerned about this. In fact, it was Dr. Begley's paper in Nature that I think got their attention initially. So I'll just leave it at that.
I think the discussion has really opened up this whole issue of the case for standards. And let's just say we all accept, hallelujah, there's a case for standards. But now we have to face the issue of how we're going to make it happen. And the issue of standards and the quantity of standards in the various areas is sometimes thought about as an ocean. And one of my favorite expressions, of course, is don't try to boil an ocean. So I'm asking the panel just 2 points. How do you prioritize what needs to be done? And how do you make the process more efficient? Because as my colleague Yvonne Reid said, it took us almost 2 years to make one very important standard. So I would like your thoughts on that, on those questions.
I guess I'll start out with that one. It's going to take time any way you do it. And if you're talking about procedural standards, consensus takes time to develop. So there's the codification of best practices and then gaining consensus throughout the community that those are indeed the best practices to be used. So that's always going to take time. We in CLSI, we're continually looking for ways to speed that up, but my guess would be that still, from start to finish, it's probably a 2-year process by the time you develop the standard and get consensus on it. So I think any way we look at it, it's going to take time. The longer we wait, the further behind we'll get, of course. So it's important to get the process started, to build that educational foundation, to get the buy-in in the community as rapidly as we can. Not necessarily an easy thing to do, but you already have people that have said, we all agree that it's necessary to do it. So there's a core group that understands it is very important. And I would expect the core group is going to work to get that moving. In terms of prioritizing, that's, too, I think, a very difficult question. When we look at material standards for the clinical laboratory, what most people, including physicians who act on the results that they get from the laboratory, don't understand is a very, 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 very small fraction of clinical laboratory assays are standardized. So if you go to hospital A and go to hospital B on the next block, and they're using different manufacturers' products, you're not necessarily going to get the same answer on the same sample for the same component. So that was the impetus that led to AACC to spearhead establishment of the International Consortium for Harmonization, is that there's many independent groups throughout the world who are doing bits and pieces, but it's in an uncoordinated fashion. So somebody in Germany may be working on standardizing creatinine while an effort is going on in the US and a third effort is going on in Japan, and that's wasted effort because you have 3 groups working on the same thing when they could be working on 3 different key things. So part of the question we're grappling with for the consortium is that whole question of priority. And we decided priority was going to be based on the health need and what the results are used for, how often that kind of test is done. If you have something that's used in an emergency situation like a cardiac marker, the importance of that being standardized, because there you're really talking time is life. That rises right to the top, as opposed to something which is more of a wellness indicator, if you will. And while it's important that that be standardized, if the results are off, they're not going to impact life. So that's one approach that we've taken in that situation.
I think for the authentication of cell lines, what actually went into that was that it had to be a very simple procedure. It had to be very low cost. And most importantly, it had to be available to a wide variety of scientists. So in other words, anyone could have access to it. And so if you meet all those criteria, then the likelihood of using it is greater, because ultimately that is the goal. And so from a cell banking standpoint, being able to identify a standard that can reach a wide variety of scientists is going to be very, very useful. I think the other thing to remember is that that standard it's a dynamic process. And so when you would have put that standard in place, you have to think that it can be improved later on as new technologies come about. And so it is not just fixed in stone. Because take, for example, when we were going through the authentication of human cell line using STR, questions came up about what about SNPs? And yet, this may be the way of the future, but the point is that we don't have enough information or background information on that to see whether or not the resolution and the robustness of the assay is as good as STR analysis. But maybe 2-3 years from now, there may be enough markers available that can give us better resolution than STR. So we have to think in context that it is not fixed. This is a dynamic process, and we can actually improve on it.
Glenn. Oh, sorry, Bill, go ahead.
So one quick one. So we've talked a little bit about carrots and sticks and so on, and to make something happen quickly, it's a lot easier to get a carrot moving someone than to develop an idea that what's the best stick that we should come up with. But that's one thing, actually, while I agree that the journals are not probably the most challenged with this problem, but they might offer the easiest carrot. And that would be, you know, the Science, Nature, PNAS, Cell journals adopt a policy that now changes the entire reward structure for the people doing the research. So if we can think of very simple concepts that could be implemented, that might be the easiest way to bring a more widespread awareness to this issue. I think that's the overall goal, is that we need to just bring a more widespread awareness to the issue and how best to do that.
Glenn, you had a comment.
The way I think about this, there are sort of two components. One is standard scientific methodological procedure, and that's one standard that I'd like to see. Every experiment should have a positive and negative control. It should be repeated. It should be performed blinded. The reagent should be validated. That's kind of scientific method. Then there's another level of quality control, and that relates to the specific reagents that are used. And then we could make a list of the things that are commonplace within science today. So they range from siRNAs, small molecules, monoclonal antibodies, and so on. So I think a lot of that could actually go ahead in parallel. I agree completely we need training so that people understand how to design and interpret a good experiment, and at the same time we need to validate the reagents that are commonly used across different laboratories.
Glenn, I agree with you totally on this, and I think, using an analogy, there are basic food groups that one should follow in nutrition. There are basic ingredients that chefs need in order to cook most of everything. The vegetable stock, the sauce. Again, going through that process you described, using a consensus approach, identifying which are those basic groups, then will give us the prioritization we need.
So I have a question for GSBI in this regard. So one of the tools that's used in many of the experiments today are animal models. And the challenge is, with all the various knockout mice that are out there, where do we actually draw a line with those as standards? Because, again, in certain diseases like Rett, you can take a Rett mouse and put it in a very enriched environment, and that mouse does better than the one that's not in an enriched mouse. And now when you're doing experiments with that mouse, whether it's in a deprived environment or an enriched environment, you can get completely different results. So where do you stop with all of this? So as Ray has pointed out, the chef uses many different ingredients, and when do you stop? So I would say to Ray, do we use the stockyard beef, or do we only use the beef that's actually been eating off of the range? So I would say the same thing with our animals in our experiments. We have to actually make sure that those are standardized as well, and the care for those animals is standardized, because you can change the results totally on the same genetic background, same genetics, different results based on how they're housed and the behavioral environment they're brought up in. So I think that the challenge for GBSI is, where do you say, okay, we can't, that's outside of our sphere right now, because I mean it can grow and grow and grow.
My response to that, I agree completely. I think the critical thing is that the investigators have to just declare what they did. There was a comment earlier that there's not enough information in a lot of the papers. Just tell us precisely what happened. Don't hide the details. You tell us what the details are, then you can make your decision and I'll make my decision in terms of the value of that information to take us forward. But at the moment, a lot of that critical information is not disclosed, and even just disclosing that would help us.
I want to make two quick follow-ups on that. One is Nature, I believe, is in response to methodologies has now, whereas they used to, the method section was so short you could barely write it, they've now expanded significantly online their methods sections, so you should be able to get as much or all information on procedures and methodologies as possible in response to that very point. And Steve, it's a good question in terms of where an organization like GBSI draws the line in terms of, where standards can be implemented in the sort of discovery basic research world. And I don't necessarily have the answer for you, but there was a really, I thought, very well-written and entertaining piece in Science magazine just a few weeks ago about this very issue of mouse models. And I don't know if you saw it, but it was making some of the very points that you just made.
And we have a couple of additional questions that have come in through the web. One of them goes back to the carrot and the stick. It's Dr. Begley, you had mentioned both a carrot and a stick approach. Are there any specific examples that you or anyone else up on the panel could give that would help to, reinforce that?
Well, we've heard some mentioned already. The carrot approach, I think we should focus on rewarding studies that are reproducible, robust, reliable, upon which the field can build, rather than studies that are racy and sexy and scientifically titillating. We need robust data to build the field to advance. So granting agencies should acknowledge those and reward those investigators that are producing data on which the field can rely. That would be the carrot. And the stick, Bill mentioned a moment ago, journals like Nature are taking the guidelines to authorships much more seriously than they had before, and they should and will continue to make it more difficult for papers to get published that are lacking in some of these fundamental things that we've been talking about already, the requirement for controls, for example. And I would regard that as the stick approach. People, scientists that don't comply with those sorts of what most people would regard as basic scientific methodology will struggle to get their papers published and should struggle to get their grants funded. I don't know if Bill wants to say.
Yeah, I actually view the latter as a carrot, because if you can get your paper in Nature, then congratulations. And so maybe they're both the same idea as a continuum. I think it's going to be my point before, maybe I didn't make it clear, is it's going to be easier for Nature and PNAS and Science to say, we do this and you will get a paper in here, than NIH saying, we need to make sure that your grant process has robust, validated data. I think that's going to take a lot more work.
Following up on Bill's, the ultimate carrot from the point of view of clinical and translational is that the regulatory agencies accept the standard, and therefore it improves and speeds up the regulatory process.
And I will echo that as well. FDA, from a manufacturer's point of view, you always look to what the FDA is going to have as their requirements, because the largest market is here in the U.S. And if you can't market in the U.S., the likelihood that you're going to be successful outside the U.S. gets kind of small. But FDA publishes on their website a list of voluntary consensus standards that they recognize. And as a manufacturer who's doing a submission to the FDA, if you choose to follow those standards, the regulatory process is somewhat streamlined. And FDA knows what you did because they're very familiar with any of the standards that they accept. So again, it's a voluntary question of whether or not you're going to follow those standards. If you choose not to follow them, the FDA is going to look at your submission in a somewhat different way. And they're going to be much more analytically critical, because they want to understand the methodology you used if you choose to not follow any of those standards that are available. So that kind of approach has definitely had an impact in the industry side when you're doing FDA submissions, whether it be for a drug or a device.
And I'd like to augment that. So one thing that I mentioned before, that we have a Center of Excellence of Regulatory Science and Innovation with the FDA, and I'd laud the FDA because they're reaching out to the academic community to try and help develop those standards and develop those reproducible assays and measurements that would be used in guidance and would be used to evaluate drugs and devices. So I think that's a great thing. So as long as the whole community could get involved, and if that program could be expanded to include more universities, et cetera, that would be huge, because that gets back to the education component, that if we educate people more to understand what's needed for commercialization, then we'll have a better scenario.
I think we have one more online question.
Yes, we do. Proficiency testing is a relatively mature and acceptable approach employed in the clinical laboratories. How might the concept of proficiency testing be employed in an academic setting?
Boy, that's an interesting question. Obviously, in a clinical laboratory, almost the proficiency testing focuses on all of those tests that are done very routinely, and those don't vary tremendously from hospital to hospital. So it becomes somewhat easier, I think, than it might in an academic setting. I'm not sure how you would look at the myriad of things that happen in life sciences research in an academic setting and say, how would you do something like proficiency testing?
I think if you have a core lab and you're running some routine assays, you would want to have positive controls, and so I think in that sense it could be useful in academic settings.
And that might be something that could be achieved with institutions in a given area, choosing to, for example, share samples and say, for the, we do this kind of an assay and we use this cell line.
Yes.
So send it off to a sister institution and see if they get the same kind of results. It's sort of a proficiency kind of.
Sort of,
Yeah. Interesting concept.
Are there any other questions here in the audience? Okay, well, I think that we've concluded our. I'd like to thank the panel first of all for giving us their insights, and I'd like to thank the audience that came here today to the National Press Club, and everyone in the ether world of the internet. Thank you again. We look forward to sharing the case for standards with you. We'll be distributing this online, and please visit our offices in downtown Washington, D.C. as well. Thanks again, and have a great afternoon.